Excercises of molecular basis of inheritance. So group the following as nitrogenous bases and nucleosides. So adenine, cytidine, thymine, guanosine, uracil, cytosine. So adenine, thymine, uracil, cytosine, these are nitrogenous bases. Cytidine, guanosine, nucleosides. If a double standard DNA has 20% of cytosine, calculate the percent of adenine in the DNA. So it's 30. If the sequence of one strand of DNA is written as follows, so 5' into 3', so it's coding strand. Okay, replace thymine with uracil. That's the write down the sequence of complementary strand in the 5' 3' direction. 5' to 3' direction. The sequence of the coding. If the sequence of the coding strand in a transcription unit is written as, write down the sequence of mRNA. This replaces the thymine with uracil. Okay, now which property of DNA double helix led Watson and Crick to hypothesize semi-conservative mode of DNA replication? Explain. Which property of DNA double helix led Watson and Crick to hypothesize semi-conservative mode of DNA replication? Explain. Complementarity. Base pairing. Depending upon the chemical nature of the template DNA or RNA and the nature of nucleic acids synthesized from it, DNA or RNA, teach the types of nucleic acid polymerases: DNA dependent polymerase, RNA polymerase. How did Hershey and Chase differentiate between DNA and protein in their experiment while proving that DNA is the genetic material? How did Hershey and Chase differentiate between DNA and protein in their experiment while proving that DNA is the genetic material? By using radio labeled phosphor 32 and protein sulfur 35. Now differentiate between the repetitive DNA and satellite DNA. Differentiate between the mRNA and tRNA. Differentiate between the template strand and coding strand. List two essential roles of ribosome during translation. In the medium where E. coli was growing, lactose was added, which induced the lack of peron. In the medium where E. coli was growing, lactose was added. Where E. coli was growing, lactose was added, which induced the lack of peron. Then why does lack of peron shut down sometime after addition of lactose in the medium? Explain in one or two lines the function of the following promoter tRNA exons. Why is the Human Genome Project called a mega project? What is DNA fingerprinting? Mention its application. Briefly describe the following: transcription, polymorphism, translation, bioinformatics.